Hello, I hope things are going well for you. I'm going to start our lecture 3 on tubular reabsorption. Hopefully you've had some time to go through the little videos on Jove. These were the videos for your lab on Monday, and those gave you a little bit of background on filtration and reabsorption and some of the things that take place. Today we're going to go into greater detail. So, are you ready? Let's start. Remember that this is step 2. So, what is tubular reabsorption? This is where the contents that are in the tubes are going to move from out of the tubes into the blood. So, we're talking about leaving the proximal convoluted tubule, the distal convoluted tubule, the nephron loop. So it's reabsorbed into the blood. It's moving from the tubes to the blood. And this is going to be active and passive transport. So we're going to have transportation that requires the use of ATP, and then passive transport. So there are two routes. One is transcellular and the other is paracellular. Transcellular and paracellular. So here we go. We're looking here at the tubes, and things are moving out of the tube and into the capillary system. Transcellular route. So basically the solute is going to enter at the top of the tubule cell. It travels through the cytosol and it exits at the bottom, the basement membrane of the tubular cell, and then it's going to enter the blood through at the paratubular capillaries. That's transcellular. That sounds a lot like absorption of nutrients in the gut. The second is paracellular. Para means around. So basically, these very small molecules, so we're talking water, calcium, magnesium, potassium, sodium, right? These very small molecules can get past the tight junctions and go around the cell. They go in between the cells. So here we have a lovely picture showing us how transcellular takes place. And remember that the apical membrane is going to be pointing into the lumen. And you can see it has to cross some interstitial fluid in order to get into the peritubular capillaries. If it's paracellular, you notice that it is moving in between the cells. In between the cells. Sodium is incredibly important to maintaining our electrical selves. And so we see it's the most abundant cation in the filtrate, and that transport of sodium is usually by an active transporter, and these are called sodium-potassium ATPase pumps. And basically what they do is that these pumps are going to use ATP to pump sodium into the interstitial space, and then potassium gets pumped in the other direction. the sodium is going to eventually flow into the peritubular capillaries. So if sodium enters the tubule cell, it's going to be co-transported or by facilitated diffusion through the sodium pumps or sodium channels. Active pumping of sodium results in an electrochemical gradient, and so this means that the low intra, that means within, cellular sodium levels facilitate sodium diffusion. In other words, it's going to go from an area of high concentration to an area of low concentration. And that potassium is going to leak out into the interstitial fluid. Okay, and so here we have active co-transport or facilitated diffusion through channels. And all that means is sodium is moving. So when we see movement by primary active transport, lost my place. So what we see is that when sodium's being moved, it can provide energy for other molecules, larger molecules, to be moved. And this is called secondary active transport. And basically, we see organic nutrients like glucose, amino acids, as well as some vitamins and some ions being co-transported with sodium. Stop and think about how cool that is. What about passive reabsorption of water? Because we know that wherever sodium goes, water follows. And so when sodium is moving and other solutes start moving with it, you have an osmotic gradient, and water is going to be reabsorbed by osmosis down its gradient. Aquaporins are very large channels that allow water through very quickly. And these are present in the PCT. Aquaporins can be inserted into the collecting ducts, but only if ADH has been produced and is doing its job. You remember ADH is antidiuretic hormone. So the solute concentration increases as water is reabsorbed into the blood, and so this starts a concentration gradient, and this also helps them enter into the tubule cell and the peritubular capillaries. So you can see that everything is being driven from the tubule into the capillaries. What about fats? Well, fat and some of the ions, urea, will follow water into the peritubular capillaries, and it's mainly by simple passive transport. So we look here at water, and notice that sodium's being pumped into the interstitial space, and so that's active transport. This creates a downhill sodium gradient. In other words, it's getting more concentrated, and so sodium is going to move down its concentration gradient. You see that in step 3, we have glucose, amino acids, vitamins, and some ions being co-transported with sodium. Then we have in step 4 the reabsorption of water by osmosis, and this can be through the membrane but most of it is through the aquaporins, all right? And then any kind of lipid substance can go directly through the lipid bilayer, and they're going to diffuse through the transcellular route. And then we have all kinds of ions in urea that actually come through the pericellular route. All these will enter into the interstitial fluid and eventually be taken up by the peritubular capillaries. Look at step 1. You see, well, that's interesting. 3 sodiums are pumped out and 2 sodiums are pumped in, and then, oh, they get pumped out again. Sometimes they go into the peritubular capillaries, but sometimes they're used to maintain the ion balance in the apical cells, or in the tubular cells. Do you remember that 3:2 ratio from someplace else? Yeah, you do. Oh, yeah? Electrical? See, you remember. So, there comes a point where the cell just can't transport anymore. And this is called the transport maximum. And basically, it's just the carriers are all full and they can't carry any more than they are. So when carriers for a solute are saturated, so that that solute can't be reabsorbed into the peritubular capillaries, then it's excreted in the urine. This can happen in hyperglycemia, where there's high glucose levels in the blood, and the glucose can't be taken up fast enough to be pumped back into the blood, so it's excreted into the urine. You should not have glucose in your urine. Glucose in the urine is indicative of diabetes. This is a good place to kind of pause for a minute, look over your notes, draw a line, stretch. So now let's look at the reabsorptive capabilities of the renal tubules and the collecting ducts. So the proximal convoluted tubule is the site of most of the reabsorption. Remember, we're reabsorbing. We're moving from what's in the tubules into the blood. So it's being reabsorbed into the blood. So we're looking at nutrients, 65% of sodium and water, most of the ions, most of the uric acid, and about half of the urea. In the nephron loop, in the descending limb, water can leave, but solutes can't. In the ascending limb, the water can't leave, but the solutes can. And this is helping to concentrate the urine, the filtrate to make urine, and to allow solutes to be reabsorbed, okay, to be reabsorbed back into the blood. In the distal convoluted tubule, in the collecting duct is going to be regulated by ADH. And so what happens is that ADH levels are going to cause an increase in water reabsorption. And basically what they do is that they ADH causes the collecting duct cells to allow aquaporins to be inserted into the apical membranes. So that's cool. Need to take up more water, then here you little cells will make aquaporins and insert them into their membranes and allow more water to be reabsorbed quickly. Aldosterone is going to target the collecting ducts and the distal portion of the distal convoluted tubule. And so here we have, they're going to synthesize and insert sodium-potassium channels, and then at their basolateral or their basement membrane, they're going to make and insert sodium-potassium ATPases. Remember, that is an active transport of sodium, and of course wherever sodium goes, water follows. So basically we don't lose too much sodium with aldosterone, but the daily loss of sodium would be about 2%. And you notice it says it's incompatible with life. Isn't that amazing that 2% would kill us? But you stop and think how much our central nervous system and our heart depend on sodium and potassium for the electrical current. Not good. Not good. ANP, atrial natriuretic peptide. Remember that this is released by the atrial cells of the heart, and what it does is it tells the kidneys to reduce blood sodium, so the kidneys stop letting sodium be reabsorbed into the capillaries. This decreases blood volume and blood pressure. And then we have parathyroid hormone, which acts not only on the bone, but on the distal convoluted tubule, and it tells the distal convoluted tubules to increase reabsorption of calcium into the circulatory system. That's amazing. It's just amazing. So let's look at tubular reabsorption. Notice that we start out in the cortex of the kidneys with the glomerulus. You see that 65% of the filtrate volume is reabsorbed in the proximal convoluted tubule. Excuse me. This is going to include water, sodium, bicarbonate, glucose, amino acids. Okay, but we see that hydrogen ions and ammonia ions and subdrugs then are going to be moved into the proximal convoluted tubule. In the descending loop of the nephron loop, water goes out into the kidney tissue, whereas as we come up the ascending loop, we see that sodium, potassium, and chloride are going to move out. We see that reabsorption is going to take place in the distal convoluted tubule, sodium and calcium. We also see that urea from the kidney tissue gets pulled into the ascending loop. Okay. Then we see in the collecting duct, look over on the very far right, in the collecting duct, you have water, sodium, and urea being secreted out of the collecting duct into the kidney tissue. And then we see potassium being pushed into the collecting tubule. And then also we can see in the collecting tubule an exchange of hydrogen ions, bicarbonate ions, and ammonia ions, mainly to help maintain blood pH going back and forth. I want you to study this picture understand where things are going, because next we're going to be talking about how a particular gradient is set up in the actual tissue, the interstitial space of the kidneys. All right, so here's a lovely little chart, and I think knowing the tubule segment and knowing the substance that's reabsorbed, remember that means it's moving from the tubule into the bloodstream. And then, can you tell me if it's active or passive? Okay, is it active or passive? Water is always passive. Osmosis, okay? So look at the nephron loop, water, ions. Are we talking, is it active or passive? In the distal convoluted tubules, we have sodium, chloride, calcium. Once again, are we talking about active transport or are we talking about passive transport? And then in the collecting duct, we see ions, we see water, and we see urea. Are the ions actively transported? Yeah. Some are passive. Water is osmosis, passive. Urea, facilitated diffusion. All right. Here again, passive transport.